Paralel 1. We are start to the first presentation entitled Exploring Climate Change Risks to Rice Production in Indonesia with CMIP6.
Hi, good afternoon. In this video, I'll present you our research titled Exploring Climate Change Impacts to Rice Production in Indonesia with CMIP6. This research performed by Army Susandi from Department of Meteorology, Institut Teknologi Bandung, and Angga Fauzan Pratama. Indonesia's rice supply is under pressure from two issues, a rising population and the simultaneous decrease in paddy fields area. Now, there is also another threat, climate change. We are seeing rising temperatures and increasingly unpredictable rainfall, which are negatively impacting our national rice production. IPCC has the World Climate Projections until 2100, named CMIP6. We can use this opportunity to understand the impact of different climate change scenarios to the rice production in Indonesia. With this knowledge, we can plan an effective adaptation strategy to climate change in rice production. Effective adaptation strategy matters because rice is the staple food for Indonesian and we want a stable rice availability in the market. Indonesia also have limited resource to adapt. By knowing more of climate change impact, we can prepare adaptation such as infrastructure or capacity building at the right time and at the right place. This study addressed by exploring SEM-IP6 dataset, which has one historical simulation and four climate change scenarios. We focus on Sumatra and Java because these two islands produce over 75% of rice in Indonesia. We explore maximum temperature and precipitation in the historical and future simulation. We built our analysis by integrating two key steps. One, the literature review on the CMIP6 climate projections in Indonesia and how weather affect rice. Two, collecting downscaled CMIP6 data from NASA. We then analyze the data using programming and spreadsheet to find insights. Our temperature findings are clear and consistent. Extreme heat become much more frequent as the climate change scenarios worsen starting around 2050 for both Java and Sumatra. For precipitation, the situation differs by region. Sumatra shows little change in the number of dry days. However, Java will face a clear increase in dry days. As extreme temperature leads to lower crop yields, we must prepare. We have a critical window until 2050 before these impacts become significantly more severe. The stark difference between the best and worst case scenario makes it clear adaptation is not enough. We must mitigate emission to reduce the severity of climate change. Our research highlights the need of further R&D and policy formulation. First, we need to invest in localized studies to guide regional adaptation strategies. Second, we need to fast-track the development of heat-resistant rice cultivars. Ultimately, the government must formulate policies that support adaptation and mitigation efforts within rice production. Thank you for your time and attention. We welcome all your comments and questions, and please feel free to reach us out through this email that is available on the screen, army.susandi@itb.ac.id. Thank you.
For the next presentation session entitled Smart Farming for Gurem Farming in Java Between Potential and Challenge. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello, ladies and gentlemen. I am Sofi Rindy Nurhutami from UPN Veteran Jawa Timur, and on behalf of Destidiana Malina, From UPN Veteran Yogyakarta, I am pleased to present our collaborative study with the title Smart Farming for Kurum Farming in Java Between Potential and Challenge. Over half of Java's farmers are Kurum. They are cultivating less than half of a hectare. Small land, poor infrastructure, and weak institutions keep productivity and resilience low. Smart farming using IoT, sensors, and apps can boost efficiencies and sustainability. Adapting these tools through local cooperative systems can make them realistic for smallholders. Gurun farmers are vital to Indonesia's food supply. Small-scale smart farming can improve food security, climate resilience, and social equity. We applied a semi-systematic literature review based on Prisma, analyzing technology, socioeconomic, and policy aspect aligned with SDGs number 1, number 10, and number 13. We focus on Gurum smart farming, framed a research question, reviewed and screened literature, and analyzed key themes from a local and global perspective from 2015 and 2025 working papers. Smart farming show potential in efficiency, yield, and climate adaptation, but the challenge remain like land fragmentation, high costs, low literacy, and weak institutions. This study reframed smart farming as inclusive and locally adapted, emphasizing farmer empowerment and institutional support for digital transformations. It broadened insight on smallholder smart farming, inform inclusive policy, and guides scalable innovation for Kurum farmers. We hope these studies inspire more inclusive innovation in smart farming for sustainable agriculture in Java. Thank you.
For the next presentation session entitled Endophytic and Rhizospheric Bacteria Associated with Phytoremediator of Crown Flower and Candlebush and Their Functions in Phosphate Potassium Solubilization. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Ladies and gentlemen, I'm Fabiana from Master Program of Soil Science 11 Maret University Indonesia. On behalf of all authors, I will present our research entitled Endophytic and Rhizospheric Bacteria Associated with Phytoremediator of Cornflower and Candlebush and Their Function in Phosphate Potassium Solubilizations. Cornflower and candlebush are known as phytoremediator plants that can survive in harsh environments, including those affected by drought stress and heavy metal toxicity. In the area surrounding the Putri Jempolan field, heavy metals such as lead, chromium, and nickel have been found at concentration exceeding safe threshold level, posing serious risks to soil health and plant growth. Given the situation, our study focused on exploring the potential of microorganisms associated with these plants. We aim to assess and compare the population density, colony morphotype diversity, and phosphate and potassium solubilization ability of endophytic and zoospheric bacteria isolated from crownflower and candlebush. The research is important because it helps identify superior bacterial isolates with high phosphorus and potassium solubilization abilities. These promising isolates have the potential to be developed as biofertilizer, bioremediation agent, and plant bioresistant inducer, which could also support the growth of other crops under similar adverse conditions. To address the research objective, we first conducted bacterial isolates from the leaf, root, and rhizosphere soil using the spread plant method with surface sterilization for endophytic bacteria. For statistical analysis, we used t-test and ANOVA followed by Duncan multiple rank test at 5% significance level. Phosphate solubilizing bacteria were isolated using the serial dilution method up to the 7th dilution on Pickovskaya agar. Isolates from halozones were then tested for their phosphate and potassium solubilization index using Pickovskaya and Alexandrov agar. Then we also measured plant growth and analyzed soil chemical characteristics. From the two phytoremediator plant, we successfully isolate 48 bacterial strains. Among them, 36 were identified as phosphate solubilizing bacteria capable of forming halozones. Interestingly, the rhizosphere of the crown flower showed the highest bacterial population density and the greatest diversity of colony morphotypes. We found that the highest phosphate and potassium solubilization index values were obtained from isolates associated with candlebus. Specifically, the rhizosphere isolates number 5 showed the highest phosphate solubilizing index, while root endophytic isolate number 8 recorded the highest potassium solubilizing index. This research will contribute to the finding on multifunctional bacterial isolates that support phytoremediator plant in improving soil health and metal tolerance. The bacterial isolates were also potential to be applied to other crops under similar adverse conditions. That's the end of our presentations. Thank you very much for your attention.
For the next presentation session entitled Response of Rice Lines, derived from genetic introgression of Oryza rufipogon and Oryza glaberrima to blast disease. If you have any questions regarding the previous presentation, please kindly write them in the chat box below.
Good morning, my name is Siti. Presentation, I will discuss response of rice line derived from genetic integration of Oryza rufipogon and Oryza glaberrima to blast disease. National rice production has been declining due to blast disease caused by the Pyriflaria oryzae cell. This disease is highly destructive, leading to tuberculosis. Climate change exacerbates the intensity of infection. Oryza rufipogon and Oryza glaberrima have natural resistant gene to blast disease. Integration gene provides resistance into superior varieties from resistant varieties, support national food security and reducing dependence on pesticide. Blast disease affect rice in more than one country, making it global food production. The use of resistant varieties is the most efficient and environmentally friendly control strategy. This research aligns with SDGs to Zero Hunger, promoting sustainable food security. The research evaluated 37 genotypes. This research employed the split plot design. Treatment test was applied to assess differences in resistant immune genotype. Heatmap clustering with triggering system was used to group genotype based on the resistance level. The research began with soil preparation and seeding. The pathogen inoculum was cultured oatmeal agar, medium, pathogen inoculation, and the final incubate in a dark and humid chamber. Observation were conducted 7 days after inoculation indicated significant differences in blast resistance among all genotype tested, with clustering categorized the genotype into 3 groups: consistently resistant, consistently susceptible, and specific resistant. Bencana Bali and I are 64 and continue to be highly susceptible, making them susceptible sensitive control. This appropriate utilizes to highlight species as donor of resistant gene. On Oryza gladerima contributed stronger genetic resistance compared to Oryza rubidogon. The resulting line exhibit good spectrum resistance without compromising desirable agronomic traits such as yield and grain quality. This study produced promising candidate line for breeding blast-resistant rice varieties. These varieties can enhance national food security through high adaptability. Moreover, they can contributing to more sustainable agriculture. Thank you.
For the next presentation session entitled Biochar and Bagasse Compost Organic Amendments to Improve NPK Availability in Tropical Insectisols. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello everyone, I'm Lydia Zela Wijaya from 11 Maret University. Today I will present our research title, Biochar and Bagas Compost Organic Amendments to Improve NPK Availability in Tropical Insectisol. Let's begin with the problem we are addressing. Soil fertility keeps declining due to climate change and inefficient fertilizer use. Meanwhile, Indonesia produces a large amount of sugarcane bagasse, but most of it remains unutilized. Compost and biochar actually have a great potential, but the residual effect, especially in the third planting season, are still unknown. From this challenge, we can see an opportunity. Bagasse compost and biochar offer a promising solution. They transform agriculture waste into organic amendments that improve soil structure, enhance nutrient retention, and reduce fertilizer losses. This creates add value while supporting sustainable farming. So why is this important? Improving soil fertility means improving crop productivity and farmers' income. Using bagasse compost and biochar reduce agriculture waste and support sustainable nutrient management. In short, healthier soil lead to better farming and better livelihoods. To addressing this problem, here is our approach. We integrate organic amendments, compost, biochar, and biocompost with reduced NPK fertilizer. Compost improves soil structure and supplies nutrient gradually. Biochar enhances nutrient retention, while biocompost combine both function. This system maintains yield, reduces fertilizer dependence, and supports sustainable agriculture. Now, let's look at what we actually did in the experiment. We applied the organic amendment once at the beginning, while NPK fertilizer was applied gradually. By combining this treatment, we evaluated how compost and biochar continue to influence soil plant NPK availability in later seasons. Data were analyzed using ANOVA and DMRT. So what did we find? Residual biocompost combined with 100% NPK resulted in the highest nitrogen, phosphorus, and potassium in soil and plant tissue. Even when the NPK dose was reduced to 50%, biocompost maintains similar nutrient uptake. This means chemical fertilizer can be reduced without lowering nutrient absorption. So what difference does this make in real farming? Biocompost increase fertilizer efficiency by retaining nutrients longer and reducing nutrient losses, integrating inorganic and organic fertilizer, improving nutrient management while lowering dependency on chemical fertilizers. And finally, here is the contribution of our study. This research proved that chemical fertilizer use can be reduced without decreasing nutrient uptake or soil fertility. Residual biocompost improves nutrient retention, support cost-efficient fertilization strategies, and contribute to long-term soil health and sustainable agriculture. Thank you for your attention, and I welcome any question. Thank you.
For the next presentation session entitled Mulching Techniques, Improved Yield and Water Use Efficiency, a meta-analysis from Bangladesh. If you have any questions regarding the previous presentation, please kindly write them in.
Welcome to my presentation. This is Sumaya Akhtar from Sylhet Agricultural University, Bangladesh. I am glad to share my study that I entitled as Mulching Technique Enhance the Yield and Water Use Efficiency, a meta-analysis evidence from Bangladesh. Here is the concept of mulching. Mulching is a simple yet powerful technique in agriculture. It helps mitigate the rainfall and the rise in the global temperature and the soil degradation, all of which are increasing due to climate change. So, all of these problems can overcome by using the mulching techniques. Now, let's look at the broader scope of mulching research. Mulching improves the soil moisture by reducing the evaporation. It enhances the water use efficiency, meaning plants make a better use of available water. It increases the crop yield. It reduces the pressure on the water resources, lowering the irrigation demand. Let's know why is this study important. Our meta-analysis combines the results from multiple mulching studies across the Bangladesh, creating a comprehensive national overview. The evidence can help the policymaker and the farmer adopt the climate-smart and sustainable farming practice under the changing climatic conditions. Here is the approach of our study. The meta-analysis approach allowed us to statistically evaluate the effectiveness of mulching across the various crop and environmental conditions. Therefore, we integrated data from 200 published papers conducted across the different regions of Bangladesh. Now let's look at the workflow of our study. In the diagram shown, the steps of our research design, from data collection to statistical analysis, are illustrated to show how statistically the study was conducted. By synthesizing these collected studies, we aim to quantify the overall effect of mulching on crop yield and water use efficiency. Here is the finding of our study. Our analysis clearly shows that mulching significantly increased both crop yield and water use efficiency compared to no mulching condition. In the chart, you can see mulch greater than zero line. All data point above it indicate positive effect across the different crop. This confirms mulching consistently increased the yield and water use efficiency. The impact of our study is that it will provide evidence-based recommendations to enhance the productivity, optimize the water use in crop productions, and support the sustainable agriculture practice under the changing climatic conditions. The contribution of our research will be that it will highlight the regional variation, showing that effectiveness of mulching can differ depending on the crop type and location. This insight can help tailor mulching practice for different regions and guide the policymaker in promoting climate smart solutions. To conclude, mulching is not just a soil cover, it's a climate adaptation tool that enhances the productivity and the water use efficiency. That is all of my presentation. Thank you very much for your attention. If you would like to ask any question, please raise your hand and we will answer your question.
For the next presentation session, entitled Conservation Agriculture Enhanced Crop Productivity by Optimizing Resources in South Asia, Evidence from a Meta-Analysis. If you have any questions regarding the previous presentation, please kindly write them in the chat.
Hello everyone, welcome to my presentation. I am Mahir Sain from Sylhet Agricultural University. Our study shows how conservation agriculture enhances crop productivity in South Asia by optimizing resources, offering a practical pathway to resilience and food security across diverse agroecologies. Across many of the working agricultural zones, farmers are battling soil degradation, water scarcity, and climate stress. Evidence on CA is abundant but fragmented. Our work addresses this fragmentation to clarify where CA delivers the strongest benefits and how it can be implemented effectively. CA creates many opportunities, like resource conservation via efficient input, soil health restoration that slows degradation, and yield supports across key crops. The priority is targeted deployment, matching practices to context where returns are highest. We unify scattered local studies into region-specific insights for South Asia. The synthesis converts mixed findings into clear guidance that extension systems and programs can use to prioritize practices and investment decisions. By raising productivity and reducing yield variability, CA stabilizes food supplies. We ran a systematic meta-analysis of 300 published papers across Bangladesh, India, Nepal, Pakistan, and Sri Lanka. We harmonized outcomes and quantified the overall effects of CA practices to enable fair cross-context comparisons. We applied standard inclusion criteria, extracted effective sizes, and used robust meta-analytic models with heterogeneity checks. We examined country, crop, and practices as moderators and performed sensitivity analysis to confirm the stability of the results. The study confirms that conservation agriculture is a key strategy for building resilience. It boosts yields, saves water, and while its impact is positive everywhere, it is tailored by location and crop type. Our synthesis offers evidence-based guidance for South Asia. CA can rise productivity and sustainability together. The takeaway for implementers is regional adoption, select practices that fit local conditions, and monitor outcomes. CA drives sustainable farming and offers a practical path to a stronger food security and climate resilience. Grounded regional evidence, practice-level effects, and policy-ready insights. In short, practical guidance for smarter local actions. Thank you. For any question, you can contact me at my email address.